Hello, welcome. My name is Mel Rutherford. I'm the chair of Psychology, Neuroscience, and Behavior. The Department of Psychology, Neuroscience, and Behavior is located on lands that have been visited and inhabited by many peoples, including the Haudenosaunee, the people of the Longhouse. The Haudenosaunee Confederacy is made up of the Mohawks, the Oneidas, the Onondagas, the Cayugas, the Senecas, and the Tuscaroras. The Haudenosaunee Confederacy is the oldest democracy on earth. The Confederacy values shared governance and has the intention of hearing all of the voices in the community. Women are a large part of the leadership in the Confederacy, and the clan mothers represent the people and listen to the voices of the people. Today, there are over 100,000 Haudenosaunee people living in the traditional Haudenosaunee territories. And today, wherever you come from, you're welcome here.
Thank you, Mel. I specifically asked Mel to do our land acknowledgement today because I learn something every single time he does that. So thank you very much. My name's Dr. Kathy Murphy. I want to welcome you to the 29th Brain Awareness Week talk here at McMaster. Our very first talk was given by Jack Diamond. He gave it to a whole room full of high school students. And we've had quite a range of speakers. They always address timely topics. Sometimes they're controversial. They are always broadly applicable and so of interest to everyone studying the brain and beyond, those interested in other areas in science. Of course, our 29th speaker aces all of those criteria. I'm really pleased to be able to introduce our speaker today, Dr. Liisa Galea. And Liisa, some of you may know her work already, the students that have had a chance in the last 2 days, and my colleagues that have had a chance to interact with her. You already know Liisa is a renowned neuroscientist in the field of sex differences, and I think very importantly is addressing women's brain health, and not very many researchers are doing that. Liisa is a Canadian neuroscientist, yay. Undergrad from Queen's, graduate degrees from Western with Doreen Kimura and Peter Osinkoff, and then off to UBC. Did you go? No, you went to Columbia? Rockefeller, yes, for a postdoc with Bruce McEwen. Right, so this incredible lineage in sex differences, and has recently moved from UBC to CAMH, Toronto. So hopefully we will have lots of interactions with Liisa now that she's nice and close. In addition to her research, Liisa is a leader in the field. So she's blazing the trail for, identifying those opportunities for neuroscientists to study sex differences, and really for reinforcing in us why it's important to do that. Liisa's got a long list of awards, president of this and that, and lots of, not blah, 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 really important stuff, but you're not here to listen to me, you're here to listen to Liisa. So join me in welcoming Liisa Galea.
Okay, this is my head is going to be like this by the, we're going to have a little bit of a crick. And did I call this talk something else? Because I realized I didn't change the title. But thank you for that lovely introduction and that really lovely land acknowledgement. Can you hear me, by the way? I can't tell if you. I need to do my own land acknowledgement because I want to acknowledge that most of this work was done in Vancouver, Canada, which is situated on the traditional, ancestral, and unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And now, of course, I'm in Toronto, which is covered by Toronto Purchase Number 13 in 1805 with the Mississaugas of the Credit, as they were known at the time. And CAMH is committed to reconciliation, and I'm excited to contribute to that. So I am going to talk quite a bit about sex. And my dad gets excited when I say that, and then he finds out what I really talk about, and then he's not excited about it anymore. But when I'm talking about, I think it's really important to define what we all mean by sex and gender. So I'm going to be talking about the biological or biomedical characteristics between males, females, and intersex individuals. Gender, sometimes people conflate with sexual orientation. It's not sexual orientation, it's how you identify as a woman, non-binary, gender diverse, man. And it's much more than that. It's how society has expectations and attributes, and gives you attributes based on your gender identity. And that is society from all different levels, from home life all the way to just going to the mall and purchasing some shoes. And all of that can have impacts on our health outcomes and disease risks. And this is my spouse, who identifies as a man, who is showing you what is expected of him in terms of his role in my own household. Neither one of these terms are binary, but they're often treated as such in the literature. And I'm going to be talking mostly about the disparities between males and females and men versus women, and I apologize for that, but I want to make it abundantly clear that all of the disparities that I'm talking about are many-fold greater in people of color, Indigenous, gender diverse, and trans individuals. And each of those communities deserves recognition and research in their own realm. And I put some really fantastic researchers that do that kind of work, but I'm also happy to answer questions about that. I'm going to use pictures from my own family because I think it's really obvious. And I'm also apologizing for the people that heard my talk yesterday. The first 10 minutes is about the same, and the last 5 minutes is about the same, but the middle is different. So I think you can very easily see some biological and maybe social gendered effects just based on pictures. And you're probably very well versed that human females are more likely to live longer than human males, but you may or may not know that they're also more likely to live with chronic disease. And this is my mother, who suffered from a severe form of Parkinson's disease, endometrial cancer, as well as arthritis towards the end of her life. I really appreciated this study that came out that talks about some other factors, but it's using the Danish registry. Came out about 5 years ago now. And buried deep in a supplemental table, they talk about 1,300 different diseases and show delays in diagnosis. And it turns out if you average all of those 1,300 different diseases, human females are diagnosed 2 years later than human males are on average, over 1,300 different diseases. Now, I think there are reasons both on the biological and on the social side of things for why this disparity occurs. But one of the major ones, I believe, is because most of our medical knowledge, including our diagnosis criteria, have been based on males, and pretty much solely males. So much so that often symptoms in females are called atypical. And you see this atypical label across disorders where there's a male prevalence, across disorders where there's a female prevalence, but you still see the label of atypical. And also in disorders where there's no sex difference in prevalence, suggesting to me that there is a systemic bias. And I think we have to think about this quite a bit because words matter and they can be hurtful. And many of us have experienced dismissal of symptoms in our own doctor's offices. And so how this plays out in terms of the medical field is really important for us to acknowledge and think about. And if you don't, I know some of you are here for a required course. If you just need one thing, this is it. Males can't serve as a default for females. There are just too many differences that we see at all sorts of levels, and I'm going to go through some of them. To me, if we're relying on what we understand occurs in males, it's like we're using the wrong playbook. And to me, it's akin to trying to fix a refrigerator with an oven manual. Some things are going to be the same, like you plug it in and it relies on electricity and there's a power button, but some things are going to be completely opposite, like it keeps things cold and other things keep things warm. As a neuroscientist, of course, I'm interested in what do we see sex differences or influences in the brain, and there are a number of them. In gray matter volume, But I want to just point out there are just as many areas that are female greater as there are male greater. Studying sex differences doesn't automatically prove that one brain is inferior to the other. We'll come back to that. There are also some putative white matter differences as well. So males being more intra-hemispheric, having more intra-hemispheric connections, females having more inter-hemispheric connections. These may or may not contribute to the fact that we see a number of sex differences in prevalence of disease. And I've put some common ones out there. One thing you might also notice from that brain cloud is that the ones that are in blue tend to show up a little earlier in life, whereas the ones that are in the pinker hue tend to show up a little bit later in life, post-puberty. And that the idea of looking at sex across a lifespan is a really important factor. I actually think it's more interesting to look at the manifestation of disease and disorders and how they may differ, not just prevalence or a difference or influence in the brain. And we see a number of these differences in manifestation. Again, both in disorders where you see a difference in prevalence and sometimes even we don't see any difference in prevalence. And the two disorders that I've been concentrating on in my own research is Alzheimer's disease and depression. And today I'll be talking about depression. So hopefully I'm hammering home the point of why I think it's so important to look at sex differences in disease, right? Because it can give us more clues on how a disease develops, manifests in that treatment of those disorders. It also allows us to build better models of disease, and that's true from both a clinical and a preclinical perspective. And better models are just going to give us better precision therapeutics. And obviously, if that doesn't convince you, our federal funding agencies are now mandating incorporation with different levels of mandatedness and different levels of success. And I'd really love to answer questions about that at the end. If you see a sex difference in your research, that should automatically cue you to think that there could be a number of factors, and I haven't put an exhaustive list out there. But biologically speaking, you think about sex hormones, sex chromosomes, or some combination of the two of them. And just so we're on the same page and I get to show an adorable picture of my adult offspring, when I'm talking about sex hormones, I'm talking about ovarian hormones like estrogens in females and testicular hormones like testosterone in males. Obviously, we have each other's hormones, they're just at different concentrations. And these hormones work on receptors that are located across the body, not just in the reproductive tract, right? They're all over the brain in many different areas. And of course, it's much more complicated than that. Testosterone itself can get converted to a very powerful estrogen called estradiol, or a very potent androgen called dihydrotestosterone. And sex hormones themselves can affect risk, symptomology, as well as treatment. It became very rarely studied. I'm giving you one example from the schizophrenia literature, just showing across the menstrual cycle, as estradiol levels are declined, psychotic symptoms increase. I really think it's important for me to talk for 2 minutes about what sex differences is not. It's not sexist. It's not more complicated in one sex than the other. It's not, this doesn't mean that males and females are polar opposites, and it's not the final step. So what do I mean by that? First of all, every single one of us in this room and every single one of us in the world has biases and notions, and it's just important to recognize them. And if somebody points out a bias to you, don't get mad at them. It's like armpits. If your armpits are smelly and somebody tells you your armpits are smelly, you say, oh, sorry, my armpits are smelly, and you clean them up, right? So it's not a shaming thing. It's just something we all have. We all have armpits. One notion is that a sex difference has to mean it's a dimorphic, that it's opposite. No, sometimes that happens, but that's a fairly rare occurrence. Another is that a sex difference in a brain means that automatically that female brains are inferior. First of all, I'm like, why is it female brains? Why can't it mean that male brains are inferior? I've never understood that one. That one really gets me. But that's an empirical question. If you see a gray matter volume difference or any kind of difference, it's an empirical question. What does that mean in terms of function? Does it mean anything in terms of function? It might not mean anything. It might be serving to actually keep the behavior the same. And we'll talk about some of these examples. The other one is that females are atypical. They're not atypical, they're just different than maybe what's seen in a male standard. And the fourth one is that females are more variable because of their hormones. And Rebecca Shansky's written a really lovely article about that. There are meta-analyses out there in rats and mice across a wide variety of physiological traits, neural traits, and also one now in humans, showing there's absolutely no sex difference in variability. You cannot say that one sex is more variable than the other. That cannot be your excuse. Now, when I see what people have interpreted this as meaning hormones don't matter, I'm going to show you some data to know hormones matter. It's just that they're not, the variability within each sex might be hormone driven, right? But that there's no, you can't say that, I can't study females because they're too variable. That's just not true. And I really like showing this data. So this is testosterone levels in human males and human females across age. And hopefully what you can see pretty clearly is that, and there's across the day, that there are very large fluctuations in testosterone levels in human males. They differ as much as 50% across the day. So given that there are diurnal fluctuations in testosterone in human males and monthly fluctuations in human females, I have one question for you. Who's more hormonal now? All right, so there are many types of sex differences. I don't want to talk about the first three because they're boring in my mind. The ones that I think we need to talk about are these hidden ones. So that might be when you don't see any evidence for, both of these times, you've got mechanistic, latent, sometimes they're called something different, convergent, divergent. But you might not see a sex difference in a trait you're interested in, whatever it might be. And I see this a lot in the literature. Oh, we didn't see a sex difference in our first experiment, so now we're just using males. Of course, it's always males for the rest of the experiments. The issue is that you often, often. I'm just going to say often. It's my talk. I get to say what I want. Often you see sex differences in the mechanisms, and that can be in molecular and neural mechanisms underlying that trait. The other part might be that you don't see a sex difference in a trait that you're interested in, but that doesn't mean that something doesn't come along like age, genotype, stress, experience that might not also elicit a sex difference in that trait. So I'll give you some examples of that. And then the final point, and you're going to hear this a little bit more in this talk than in the last talk, is that women's health is much more than comparing to men's health. So there's this idea that, because of these disparities we see in women's health, SGBA, or sex and gender-based analysis, is the answer to overcoming all of these disparities. It's only going to get us part way. To me, that's just like sort of the first step, the first clue that there might be something going on here. We already know that female-specific experiences can drive health outcomes and disease risk, and all deserve study. But you'll see at the very end, very few people are doing this kind of work. So today, I want to give you some clinical examples of sex differences in major depressive disorder, some preclinical work that we've done looking at a cognitive trait of depression called negative bias. And then I'll spend some time talking about what we can, sort of single-sex studies, and what we can learn from piecing apart the heterogeneity of perinatal depression. And it'll come to why I want to talk about that in just a bit. No matter who you are or what disorder you're looking at in your own research, it's really important for you to understand who's getting the disorder and when, because that really can give you a lot of clues on the etiology of that disorder, and then maybe even treatment. So when we're looking at this I really like this particular graph. It's across the lifespan, female to male ratio, and where it comes across above 2 is really the sort of reproductive years, people call it. And that suggests that there's a unique physiology in females that might incur the susceptibility to depression across their lifespan. So why not use that information to understand the disorder more? It also leads to perhaps 2 alternative biological explanations. One being that females are more susceptible, and I'm a neuroendocrinologist, so I'm always going to think hormones are at the basis of it. So due to their fluctuating hormones, and males are more resistant to the depression because of their hormones. And we've created animal models looking at all of this. I'm not going to talk about all of that today, but I'm, again, happy to answer questions about this. And another question we have been interested in, and I'll show you a tiny bit of this data, was, does antidepressant efficacy, does it change based on the hormonal status of the, in this case, animal, under different animal models of depression? Now, I get asked this question all the time, so I think it's better just to head it off. And that is, don't men and women just show depression differently? So let's look at our diagnosis of depression. To be diagnosed with major depressive disorder, you have to have one of those 2 blue symptoms and 5 out of the other 7 symptoms for at least 2 weeks, and it has to interfere with your life. I think if you're going to study this, the best last name ever, right, could be this one. It really, 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 really hard to argue with this N, right? Over 78,000. This is in-person interviews, door-to-door knocking, door-to-door, as well as going to communities. Here's some do a little survey for us. 5 different countries in Europe, so it is a European sample. But it wasn't until it was 5 or more symptoms that you start to see this increase in the ratio. Now, depression's really difficult to model clinically, preclinically, right? Because you have so many heterogeneous symptoms. You've got weight gain, you've got weight loss, you've got insomnia, hypersomnia, you've got agitation or retardation. Like it's a little bit of a, I'm going to say a word that I can't say out loud because somebody's live streaming me right now. It's complicated, right? It's complex to model. And there are other symptoms as well that can come into play. And in fact, Bush and Liston, maybe that's coming up, which you can find with that QR code, determined that there are 256 unique symptom combinations. That's a lot. So, even when we talk about animal models and how they're not good enough for human disease, I think we don't also understand human disease as well as we could, right? So it's an interplay between the 2 of them, I think. Are there sex differences just in the symptoms of major depressive disorder? Really hard to find this data, really, really hard. But there are a few. So there seems to be more hypersomnia, more hyperphagia, more atypical symptoms, my favorite, and some more rumination in females and in males. I started thinking about this atypical a little bit more, and I started thinking about this in terms of depression. And this, I came across this particular study, which is a really fascinating exploration of these 7 different scales that people use to diagnose depression in their work. And he found very little overlap between the 7 common scales. So here are all the symptoms that are looked at. And this is really busy, so I appreciate that. But each of these, so it's got the 7 scales, got all the different symptoms, and you can look at the pretty colors there while I'm talking. If it's a solid circle, it means they directly asked about that symptom. If it's an open circle, it was an indirect question about that symptom. And then I started thinking, well, if females have these atypical symptoms, which scales are asking about those atypical symptoms? And then, I didn't do this, exhaustively, but you can start to see actually the scales are not asking about those questions. So actually, are we missing more people than we know about out there? But what about biomarkers of depression? Hippocampus volume, one of my favorite regions, as some of you know, is the hippocampus. We know from meta-analyses that there is a smaller hippocampus. Stress system is perturbated. Again, meta-analyses show higher levels of cortisol and impairments in negative feedback. Immune system, higher levels of pro-inflammation, higher levels of metabolism. Are there sometimes you just have a simple question, are there sex differences in biomarkers? Super hard to find this. Super hard. I would love you to go and find me more. I'm sure there are a few more than I've found here. But the ones that you do, because people aren't looking at it. So either they'll use it as a covariate or they'll have a mixed sample, but they won't actually use sex as a meaningful variable in their analysis. So it's really hard for us to understand, are there sex differences in the biomarkers? And if there are, we need to figure out what they are and what direction they're in. Okay. I'm going to give you a really good example of why this matters, because I do see some people raising their eyebrows at me, probably for other reasons. But these papers came out about 5 years ago or so now. This is postmortem tissue transcriptomics, 2 different labs, finding very little overlap in gene expression from people with major depressive disorder, males and females. Very little overlap. And then Marianne Sene and Etienne Sibyl, who were at CAMH, found that even in the genes that were overlap, which are shown here, look at that. They go in opposing directions. So even in the genes that are overlap with major depressive disorder in men versus women, they go in opposite directions. So again, really important for us to understand this. And I think is a very nice example of a mechanistic sex difference. Okay, if you look at common risk factors for major depressive disorder, female sex, I already told you, comes up pretty high. It's not the highest, but it's pretty high. But other things like chronic illness are high, family history, and things that I'm going to call chronic stress come up. And so in my lab, we look at the interaction between female sex and chronic stress. That's how we're trying to model depression in our lab. And I know many of you are very skeptical. If you don't do animal work, how could you possibly model this, right? And I would say that most of the animal models that are out there are based on either chronic stress. If you see some on acute stress, which you do, I don't personally think that that's necessarily a good, I'm going to get the word wrong, necessarily a good ecological, not ecological, ethological, whatever, a good model of depression, because acute stress isn't what seems to precipitate depression in humans, right? It's chronic stress. The other models that are out there, there's a few of them based on sex hormone withdrawal, which I'll talk about. Of course, it's really hard to model depression in animals because we can't ask them about their thoughts of suicide. Well, you can, but they're not going to. But we can absolutely easily look at all those italicized symptoms as well as the biomarkers. And in my own lab, when we're looking at this, we don't look at this in every single study that we're looking at, but we do tend to look at a variety of behaviors. We look at the endocrine pathways. We are branching out from the hippocampus, but we do look at a lot of features of the hippocampus. I'll talk about that. Look at inflammation as well as other features as well. And actually, we've been criticized for this. Like, that's too many things to look at. But when we're looking at humans, we're not asking them one question on the Beck's depression inventory. We're asking them many questions. So I find that a really interesting comment. As I've mentioned, I'm interested in the hippocampus. Why? We know from work from Shailene that with untreated depression, the longer you're depressed, the more likely you show up with a smaller hippocampus. So it suggests that there's integrity loss with major depressive disorder. It's also a brain region that shows a lot of hormone receptors and also a very large concentration of glucocorticoid receptors, which is the main stress hormone receptor. And it's very plastic in adulthood. And there are a lot of different ways to look at plasticity. One of the ways we do so in my lab is looking at the production of new neurons, which I'll talk about more in a bit. But I really want to talk about this region here, is CA3 region, because, as was mentioned, my postdoc mentor, Bruce McEwen, who died just before the pandemic, pretty suddenly for us, but I would say he's like sort of like the father, maybe grandfather, no, whatever, I don't know. He's really important, I think, in the stress literature, and did sex differences far before I started looking at it. But one of the things he had found in his lab was that if you did a chronic restraint stress, it's 3 weeks, he found apical dendrites were atrophied, so they sort of shrunk. And when I came to his lab as a postdoc, he was very, very nice postdoctoral mentor, and he said, yeah, sure, you can do it in females if you really want to. He didn't say it like that. He was like, yes, of course, you should do that, Lisa. So I did the study, and we found that, no, there was no apical dendritic atrophy. It was basal. And I'm sure many of your eyes are glazing over right now, saying, that was really, really boring. Why are you telling us this, Galea? But I'm telling you this because, for one reason, because this apical atrophy is associated with poor performance in spatial mazes in males. It's actually associated with facilitated performance in females. So this idea that a reduction in gray matter is always going to be bad isn't necessarily the case. So in terms of neurogenesis in the adult hippocampus, there are many different components to it. I'm not going to go through all the components, but you can visualize these cells in many different ways using endogenous markers, or if you're looking long-term, you'd want to look at an exogenous marker like a thymidine analog, like bromodeoxyuridine. And that just tells you there's a new cell. It's a thymidine analog, so it's just going to get incorporated into any new cell that's dividing. If you want to determine what kind of a cell it is, you have to look at another kind of marker, like a mature neuronal marker like NUN. And then it might not be the number of these new cells, but how active are these new cells. And you can look at activity of these new cells using immediate early genes like ZIF268 or CFOS. And after 40 years or so, hippocampal neurogenesis seems to play a role in stress resilience. There's some controversy, but also pattern separation. And I'll talk about that in just a second. And many of these new cells, there's been a lot of emphasis in the literature because a lot of these cells are produced, but most of them die off. And so what seems to increase cell survival or what decreases cell survival? And I put a number, this is not an exhaustive list that are up there, but I just want to say that sex plays a role in each of those factors and also plays a role in the etiology of depression. So we had a very simple question many years ago, I guess now, are there sex differences in neurogenesis? And there aren't any. So this is in young adulthood, and this is in middle age. And you can see a huge decrease in neurogenesis that's shown across a wide variety of species, but there's no actual sex difference. But we had some thoughts that maybe the maturation process would be quite different. And so Shunya Yagi, who just finished his PhD in my lab, gave a DNA synthesis marker, then perfused the animals several times later to look at this question. And so he's looking at these BRDU cells and how many of them also co-express mature neuronal marker NUN across time. So I've got 1 week and 3 weeks I've shown you here. Across time, this increases. That's what everybody sees. By 3 weeks, this is in rats. About 80% of those cells will co-express mature neuronal markers, indicating they're new neurons. But at 2 weeks, it's the males that show more co-localization with these mature neuronal markers, suggesting they're maturing faster. And remember, I said a lot of these cells are produced, but then they die off. Well, that's exactly what we found in our own part of the study in males, but in females, you don't see that same kind of trajectory. And I really wanted to call this paper, Is It Better to Burn Out or Fade Away? For some of you Neil Young fans. But the editor wouldn't let me. So it's called Sex Differences in Maturation and Actuation. I mean, it's just not the same ring to it. But anyway, I hope this has given you another example of a mechanistic sex difference, right? No sex difference in the three-week-old new neurons, but the pathways that they're using to get there are quite different. So does sex affect the ability of antidepressants to increase neurogenesis in the dentate gyrus? I haven't told you, but maybe many of you know that a chronic but not acute exposure to antidepressants can increase neurogenesis in the hippocampus. And this is an old study, but I put it up here anyway. It's, I can't believe 2013 is old, but it's old now. Presumably all of you were born before 2013. This is from the Stanley Medical Research Foundation's postmortem tissue. So they have these three groups, non-depressed individuals, people that were depressed that were prescribed antidepressants, people that were depressed with psychotic symptoms that were prescribed both antidepressants and antipsychotics. And this is work of John Epp, who did a PhD in my lab, is now an associate professor and a fully fledged associate professor at the University of Calgary. So these are the males. These are human males. You can see no difference across the human males. It was the human females that showed this antidepressant increased boost. And again, not that much work done out there to show you on this particular topic, but we do see that hippocampal volume is increased in female responders, but not male responders. That's kind of interesting. And we also saw this antidepressant boosting effect was only in the younger population. We didn't see it in an older population, which is the same kind of finding that Lukinson had also found. And I think this could be an example of a latent sex difference. Okay, I want to talk about some new work we've been doing on something called negative bias. So negative cognitive bias was developed by this postdoc in my lab, Dr. Travis Hodges. He's now an assistant professor at Mount Holyoke University, and he describes it in this way. It's when you take an ambiguous statement or stimuli and have negative connotations to it. So he's a very positive person, and he wears interesting shirts. And so he would say, somebody was saying to him, "Oh, that's an interesting shirt you're wearing." And he, because he's a positive person, is like, "Thank you very much." But if you had a negative bias, you'd be very upset by that comment, right? You have a negative cognitive bias. Negative cognitive bias increases severity of major depressive disorder. It's resistant to treatment. It also can predict future depressive episodes. So it's a pretty attractive thing to study. And it requires something called pattern separation. And we know that adult neurogenesis is required for pattern separation. So what is pattern separation? I think there's really good examples in real life and in movies of exactly what pattern separation is. It's the ability to form distinct representations of similar looking inputs. So it's like being able to find the green-lidded jars, similar looking jars in the pantry. And it requires neurogenesis. So the way we looked at negative bias was this task. 2 different environments. They got shocked in one task and not shocked in the other. And it was a repeated presentation, counterbalanced across days. And then on the last day, Travis gave them an ambiguous context. So half the features of the shock context, half the features of the non-shock context. And the idea was, look, if they're freezing, that should suggest that they think they're going to get shocked in there. So that perhaps they'd have a negative bias. And if they weren't freezing or didn't freeze as much, that was a neutral or even perhaps a positive bias. Travis also looked at a variety of biomarkers as well. So this is across the lifespan, adolescent, young adult, middle-aged, and there was actually no difference, kind of surprising to us, in terms of their ability to discriminate between the 2 paradigms. Maybe not super surprising, nor a lifespan difference as well, or age difference. But when we looked at how much they froze to the ambiguous context, just looking at the males first, as they aged, they got more negative, which I have to tell you, I'm now living with my 87-year-old dad, and anecdotally, I'm just going to say this checks. The females, we didn't see that same kind of increase in bias, and so the only time we saw a sex difference per se was in middle age. But these animals aren't stressed. There's no animal model of depression here. Oh, but it's also probably a good example of latent difference, right? And there's no sex difference early on in life. It's later on in life where you see this. So what about after chronic unpredictable stress? Both males and females upregulate. They both showed negative bias, more freezing to the ambiguous context. So behavior is different, so we can just go ahead and just use one sex. I'm glad we didn't do that. Because when we looked at inflammation, this is looking at a variety of pro-inflammatory markers. It's just the basolateral amygdala. We saw that pro-inflammatory cytokines were increased in the females, not in the males, not in the basolateral amygdala anyway, suggestive of a mechanistic difference. Of course, we looked at neurogenesis, chronic unpredictable stress, males and females had decreased neurogenesis, which is what you might expect to see. But it was actually only related, the amount of neurogenesis was only related to negative bias or freezing in the ambiguous context only in males. It wasn't related in females. And when we looked at activity, it was in males that showed greater activity in response, in those new neurons in response to the ambiguous context. And I know this is really, really ugly. I didn't get the altered figure in time, so you can see I couldn't figure out how to do curved labels in PowerPoint. It was driving me crazy. So I just stopped. But this is what we're calling functional connectivity. And so while I'm explaining it, just look for patterns. So you'll see like dorsal hippocampus, ventral hippocampus, amygdala, and probably you already can figure out what this is about, but it's correlations between CFOS activity between all these different regions. So these are only correlations of absolute value of 0.5 or more. Red, positive correlations, blue, negative correlations. Thicker the line, the stronger the correlation. And hopefully you can see, there are some similarities, but there are also some differences. And so our next step is to do some DREAD. We've already done some DREAD analysis in different regions to see, can we manipulate negative bias in different, does it manipulate negative bias in the same way? And of course, it doesn't. But it's not ready for showtime yet. So I'm showing you this because what I think these data suggest is that negative bias is different representation in females versus males. It looks like there's more of a neuroinflammatory signature, at least in the basolateral amygdala in females, and more of a neuroplasticity signature perhaps in males. It looks like they're at least engaging their new neurons a little differently. And there could be different neural networks that are active in response to negative bias. And I'm pointing this out because Etienne Civil and Marianne Senay have been showing the same thing in human tissue. They've also identified different pathways that seem to be affected with major depressive disorder. So I think that's one reason why it's super important to look at this. Okay, I'm now going to flip to female-only studies and keep an eye on that clock. And I won't be able to get through everything I wanted to get through. So female-specific experiences, can they influence major depressive risk? I've shown you this graph already. This suggests, if you're thinking about hormones, because I said reproductive hormones, they must be causing a risk, right? But there's actually more nuance to this story. They could also be providing resilience. Because the time of greatest risk to develop depression in a person's lifetime, is in the postpartum, the first time. That's something we shouldn't ignore, yet very few studies are looking at this. The secondary time, perimenopause. So it's both associated with a decline. And so I think postpartum is a really important time to look at. I added this joke in, and I don't have time for it, but I'm going to do it anyway because it's up here now. It's impossible right now for you to scroll through social media without seeing the latest celebrity that's pregnant, right? So you think it's amazing and gorgeous and beautiful because they're all gorgeous and beautiful. So I'm going to share with you the worst picture of me ever taken in my life, which is to prove a point that it takes a tremendous toll on a person's body to produce that baby. Pulmonary output decreases by 50%, cardiovascular output increases by 50%. There are 4 extra liters of fluid in a person's body at the end of pregnancy, and hormone levels climb dramatically. In fact, it makes a lot of sense that there could be repercussions for pregnancy based on all of these changes. And the last thing I wanted to mention, I thought was on this slide, is that hormone levels increase really dramatically during pregnancy. Estradiol, for example, is 200 times normal levels by week 20, 300 times by week 30, and climbs even more dramatically than that. And then what happens with the expulsion of the placenta is that people are hypergonadal. So they have very, very low levels of hormones, and that's for about 6 weeks. So it's very possible that this withdrawal from these not ovarian hormones, placental hormones, could be triggering depression. And pregnancy itself, we know, can alter disease risk, both in the short term and in the long term. And so yesterday I gave the talk about the long term. Today I'll talk a little bit about the short term. Wiebe Voigtjager, who's a psychiatrist in Denmark, said, I think the postpartum period is a perfect storm for depression. And I said, I love that. And now she denies she ever said that, but so I can take it, but it's hers. So all of the same biomarkers I've showed you before, they all occur in normal situations in postpartum. Every single one of them. No wonder that's a time of greatest risk to develop depression in any person's lifetime, postpartum. If you look at the criteria for perinatal depression, which is not its own little thing, it's a modifier of depression. I think that's how they call it. They say it's depression during gestation or up to 4 weeks postpartum. It's kind of a specific time point, but okay, let's go with it. If you look at depression onset postpartum, it's more likely to be associated with first-time depression. If you look at depression onset during pregnancy, it's more likely to be a recurrence of depression. That tells you, again, a lot about the etiology of these different kinds of susceptible periods. This is some work from Monk Olson showing that the relative risk of any mental disorder, and this is hospitalization, really low during pregnancy. But look at those odds ratios later on. They're really, really high, especially in that early period after. They're still really high for about 3 months. But until 19 days or so, they're really, really high. And so there's this idea that there's really, really high levels of estradiol and progesterone and cortisol and all sorts of other placental hormones when they're withdrawn could cause a depressive-like endophenotype in susceptible individuals. And so we based 2 animal models of postpartum depression based on this early withdrawal in the postpartum or increased court during the postpartum for sort of an early postpartum phenotype and a later postpartum phenotype. And I'll describe both of them for you very quickly. So this is the very first animal model of postpartum depression. I can pat myself on the back because I'm being live streamed. Whatever. I'll take applause later. So I think we published it in 2001. So it was a really simple paradigm. We just took a normal rat pregnancy. We gave them like high levels of estradiol and progesterone, and then we just withdrew them after 3 weeks. And what we found was a depressive-like endophenotype. So I know some of you won't like the forced swim test, but we can argue about it after. Okay? Increased coping in the forced swim test, a decrease in sucrose preference, which is akin to anhedonia. It's one way we can look at anhedonia. And we saw reductions in neurogenesis, so neuroplasticity. If we didn't withdraw them from the high estradiol, we didn't see the same phenotype. Or if we gave imipramine, we could rescue all of those effects. Now, what's really fascinating is that they've been doing some of this work now in humans. And one of the first people to do this was in Ruben Lau's lab, and they took people that had a history of postpartum depression, gave them a very quick hormone-stimulated pregnancy, and then withdrew them from these high levels. And look what happens on the depression scale. They really go up. But you can also see a slight increase here in the withdrawal in the control group, which is exactly what Wiebe Voigtjager showed in 2015. She withdrew younger women from ovarian hormones and then found this sort of slight increase in Hamilton Depression Score. Wiebe's gone on to look at, it was actually the amount of that decrease in estradiol that was associated with increased Hamilton Depression scores, and she's seen functional connectivity differences, so a decrease in functional connectivity to the hippocampus, for example. So I think what this is showing us is that withdrawal from ovarian hormones increases depressive-like endophenotypes in younger human females as well as rodents, suggesting that ovarian hormones are providing resilience. Well, we also wanted to look at that sort of older time period. So Rand Eade, who just finished a PhD in my lab, actually many years ago now, looked at overectomized rats and sham rats and gave them 6 weeks of chronic unpredictable stress. It's a long time. But she did this because after 3 weeks, you get a depressive-like endophenotype. And then for the last 3 weeks, she gave them a vehicle or an SSRI, Prozac, to see can we offset it. And she looked at a variety of outcomes. And this is going to be a busy graph, but they're pretty clear data. And I'll walk us through it. The green bars are the ovexed animals. And for every behavior, every endocrine thing we looked at, we saw a depressive-like endophenotype in the ovexed animals. So more passive coping, more anhedonia, more anxiety in the novelty-suppressed feeding, and impairments and negative feedback. And what we were also interested in was fluoxetine, and whether fluoxetine would have a difference based on ovex or sham animals. And it didn't work. It actually didn't work at all, not for any behavior we looked at. It works in males, does not work in our hands in females. It did seem to reset impairments and negative feedback. At least it worked on an endocrine level. So I'm going to skip this slide. So what I think this is showing us is that longer-term withdrawal from ovarian hormones in combination with exposure to stress increases depressive-like endophenotypes. And the efficacy of fluoxetine was limited to neural. We saw some neural rescue. We just didn't see it behaviorally and in endocrine outcomes, suggesting again that ovarian hormones are providing resilience. Now, I'm going to tell you it's not always true. And maybe another person wouldn't show you these data because that's a really nice story, but it's not. Estrogens are not uniformly antidepressive, and I think that's a really important and valuable message. And I'll tell you why, and I'm going to leave Elon Musk to tell you why that's important. So just have to wait for it. So Rand also wanted to know, well, okay, so could I prevent a depressive-like phenotype if I gave estrogens? So she gave a variety of ER alpha, ER beta, or estradiol, and then gave them chronic unpredictable stress. And we switched species. These are now mice. And what she found was that ovexed animals here in this gray bar here increased passive coping in this is a tail suspension test. I always want to say trio social stress test. The tail suspension test, because forced rim test doesn't work as well in mice as it does in rats. So ovex did the same kind of thing that we thought it would do, and estradiol, look, it reduced passive coping, so that's great. And all of the agonists also decreased passive coping. It's a really nice story. I don't know if you noticed, this is in the non-stress group. In the stress group, we didn't see any evidence that estradiol actually protected them at all. And we are going to borrow from the healthy cell bias action of estrogen. That's Roberta Brinton first proposed this. And so we think that estradiol in a healthy brain is doing great things. But in an unhealthy brain, a stress brain, It's just not providing any kind of efficacy. And now we did see that estradiol did provide some efficacy on inflammation, but it was just in the hippocampus. I know I'm going through that quickly. So again, estrogens are not uniformly antidepressive, as they're not providing complete resilience to chronic, unpredictable, stress-induced endophenotypes, depressive-like endophenotypes. But a loss of ovarian hormones, like in the postpartum period and the perinatal for longer periods of time, do seem to drive depressive-like endophenotypes. And I told you Elon Musk was going to come into this. I don't know if you know, but a month ago, he tweeted this out: hormonal birth control makes you fat, doubles risk of depression, and triples risk of suicide. So there's clear scientific consensus, but very few people seem to know it. And that's because there is not clear scientific evidence. I know, fancy that, right? And we'll have a whole session on this in a conference that I'm doing. Yeah, I know. It's really dangerous, right? That's really dangerous. Anyway, here's the data that first suggested this. This is antidepressant use, not depression. In the earlier years of life, it does seem to be associated with more antidepressive use. This is in Denmark, with a variety of hormonal contraceptives. But in meta-analyses in the adults, using a variety of contraceptives do not indicate any increased risk. I mean, I still think this is really interesting. If in earlier ages, that's what's driving it, why is that the case? And we can talk about why that might be. Okay, I have far too much in the time we have left. So I'm just going to end on I'd love to tell you about perineural nets. Wasn't there somebody that wanted to hear about perineural nets? How much can you see? Don't look at how many slides I'm going by. Don't do it. Don't look at it. Don't look at it. Don't look at it. I know it's so good. I wish I could tell you more. I'll tell you a few more things, because I can't not talk about brexanolone. I can't tell you. And one reason I want to tell you about brexanolone is it's the first time the FDA approved a drug for postpartum depression. And why did they do this? They did this because Jamie McGuire looked at a model of postpartum depression, and she found that allopregnanolone, which is a metabolite progesterone, seemed to alleviate depressive-like behavior in her mice. Lo and behold, flash forward a few years later, they do clinical trials and they find that it works for postpartum depression as well. That's a good news story. And I think we need to think about that. I want to show you one other thing. I don't want to show you that because you're going to get mad at me. Okay, so I think that, the fact that I'm talking about major depressive disorder, and then I flip to like a subset of major depressive disorder, which is perinatal depression. I say, hey, there's heterogeneity, and we should really think about that. I really think it's an unmet need for lots of things. And it's affecting us maybe negatively, right? Because if we don't pay attention to this, I think this has significant repercussions. So I already told you that, you see de novo depression in the postpartum. That's 40% of cases. So maybe that's why people didn't want to necessarily take a look at it. Premenstrual dysphoric disorder, 80% of the cases, the depression happens in that first week, prior to menses, that premenstrual week. But 17.5% of people actually show it all throughout the luteal phase. So instead of hiding behind that heterogeneity, I think we should explore that heterogeneity, because lo and behold, allopregnanolone, brexanolone, all those synthetic allopregnanolones, they work for postpartum depression. They do not seem to be working for premenstrual dysphoric disorder. That's why it's so important for us to understand. That's what precision medicine is all about, right? And I do apologize, I'm using only females in my data. I know it's not as exciting. But understanding what's going on in females will lead us to understanding intersex health and also male health. And ask me a question about lazeroids, and I can tell you more about male health. I'll just end by saying, we did this study at the start of the pandemic. We looked at 3,000 studies. I didn't. Rebecca Reschlin and Talon Splinter looked at 3,000 studies and looked to see whether they were looking at males and females. A lot more studies are looking at both sexes, but only 19% of those studies did an optimal design. That included, did they disclose sample size? Like I know, I read those articles. We can talk more about them. 5% use sex as a meaningful variable. 5%. So just because you're using them, it doesn't say very much unless we analyze to see if there's an effect or not. And there were 27% male-only studies versus 3% female-only studies. So let's try to correct that. Even if a few of you do it, I would be very happy. And I think that's how I should end. Is there anything else I want to say? Don't look at how many. Don't look at it. Don't look at it. Don't look at it. Don't look at it. I'll just give you my conclusions, which you already know what they are, right? Understanding basic neurobiology and disease mechanisms in males, females, and intersex individuals will lead us to better individualized treatment. I forgot how I put it. And then please, if you can, come to Women Mind Conference, April 17th. And it's 80% sold out, though, so come, because there's a whole, I have Inger coming to talk about who is at risk for depression when using hormonal contraceptives. So if you're interested in that, and there's also a session on transgender as well. I'm also president of the society. There's a meeting in Norway. It'll be in New Mexico last year. I'm so glad I'm being live streamed because I'm not allowed to say that. Hello. And please join the Women's Research Health Cluster. It's an initiative I started a few years ago, and we're really trying to spread the word, and we're trying to empower people to study this and give you tools to study it and give you the networks out there. So there's an expert database. You can look for supervisors. You can, I want to do this, but I have no idea how to start. You can find the people that can help you through that. We have blogs. We have a podcast. There's lots of ways to get involved. We also have awards. You can apply for awards. And for a long time, we had a success rate of 100% because people weren't applying. So apply. Just apply. It's no longer 100%, but it's pretty high, like 80% or something. I need to thank these people, which I did try to do throughout, and all the funding agencies, past and present, that have given me work, or money to do the work. Thank you for your attention.
Thanks, Lisa. Another amazing talk. Lots of food for thought. Questions for Lisa? I'll let you field them,
Yeah,
Lisa.
sure. I know your name. Thank you, Katrina.
Thank you for this powerful talk, and I'm especially intrigued about the effect of stress blocking estrogen's effect. I'm just wondering if you have any thoughts or if there's evidence for some neurobiological mechanisms that could underlie that.
Well, I was trying to, so I got lost with the blocking. Estrogen's blocking. You mean you're talking about like it didn't provide resilience?
Yeah.
So it did for neuroendocrine features in the neuroinflammation in the hippocampus, but for everything else it did not. So I wouldn't say that it's completely useless. It could absolutely be. It could absolutely be the dose. We only use one dose. Maybe we need to use another dose. But I think it's. We did use estradiol. I don't think it's necessarily that. You could think about the type of estradiols. It's just going to be more complicated than that. You're asking me about mechanism, and I would say one person's mechanism is another person's descriptive. So I would think, it's kind of mechanistic, but it's. I know that you want to know even more. I think that you're going to see, as we just show there, you're going to see regional differences. So, and the other thing we've been noticing is that there's a disconnect. So neurally, in general, we actually see that antidepressants are doing what they're supposed to do in females, but behaviorally they're not doing what they're supposed to be doing. So that was the other part I was going to show you. And if we tweak the inflammatory system, we do get better efficacy. It's not a complete story yet, but that might be one pathway. I think metabolism is another pathway to check, if that helps. Yes, Isabel.
So that was a wonderful talk. There were lots and lots of things
Yeah,
that
too
may have
many.
just flown by me
Yeah,
so quickly
well, sorry.
that I didn't fully understand.
No.
You sort of tried to make the argument that females often will get a disease later than males.
Yeah, they're diagnosed later.
Okay, they're diagnosed
They're not
later.
getting it later.
They're just discovering it.
Yeah.
Okay.
Bipolar disorder is 40 years later.
Okay.
women are diagnosed with bipolar disorder 4 years later. They're not getting it later.
And that's because of the atypical nature of
I
how it presents.
I think that's a feature. I don't know that anybody knows why. I mean, for some disorders, it is going to be timing differences. So schizophrenia, for example, you do see there are two sort of vulnerable time points, susceptible time points in a woman's lifetime, just in early adulthood, but also in around 45 years of age. So there are some time, and you don't see that same kind of time course difference in men with schizophrenia.
Okay, and my second question, what had to do with that birth control?
Yeah, yeah,
study you showed
yeah,
me.
yeah, yeah.
I was really curious to why taking it at earlier ages
Mm.
does predispose you to depression when taking it at later ages doesn't. What's the thinking behind that?
So no one's looking at that yet. I mean, Natalie Tronson's doing a little bit of that. We just started. Bennett is there are. See, one of the things I didn't show you was that. So I told you 3% of studies are looking at female only. If you then break it down into all-time neuroscience studies, like from whenever they first started coming out to now, it's 0.15% of studies are looking at hormonal contraceptives. 0.15%. What is the most prescribed drug in the world? Yeah, 300 million people worldwide are taking it. There's data on thrombosis, fertility. There is 0.15% of studies on brain. That's a problem, right? That's like I know, and I get a little flustered because I do talk a lot about women's health, and I do see some people getting like a little, this is boring. We need more people to not think it's boring, because it's actually a public health, could be a public health crisis. Actually, hormonal contraceptives are associated with reduced risk for Alzheimer's disease. That's good news, right? Why is that? So I can't answer your question, but we're starting to look at it, and we do see some HPA differences. That's about as much as I can say. There's huge metabolism differences between age. I mean, they've also looked at a very obvious thing is, oh, well, like I went on the pill because I wanted to, engage. I'm just like, I want that off. But that's not what it seems to be. It's not related to, sexual behaviors. It's not related to, breaking up with your boyfriend, that kind of stuff. That doesn't seem to be what's related to it. So,
Thank you.
you're welcome. Yeah, Helen.
So I know you're like a neuroendocrinologist, and you keep doing like those experiments with mice design, and you talk about how we want more models on females or intersex and stuff.
Yeah.
Have you ever done or thought of doing like a 4-core genotype instead?
Yes.
Of, like,
Everybody always asks me that, including our Arnold. And yes, we will do it. He wants me to do it with the pregnancy stuff, which I actually think would be great and shut up a lot of reviewers. I think that I think it would be really, really great to do that work. We haven't done that, and he also has another. He also has a rat model of the 4-core genotype too, and he also has another one that I think is really interesting with X dosage. So he has, not just 2 X's, 3 X's. Anyways, yeah. So I think that would be really fascinating to do. Vanessa.
I don't know if you would answer this question, or maybe your colleagues are looking at this, but you mentioned that ovarian hormones are they help for resilience of depression. So how does that interact with like hormone replacement therapy?
Yeah.
And having like somebody that's transitioning from female to male versus male to female.
Yeah.
Because the one you're introducing female hormones, and the other one you're introducing male hormones. I'm wondering how that may affect rates of depression or susceptibility to it.
It's an excellent question. So again, not that many studies out there. And it really, so the other thing that is, what also is really fascinating is that we will often say, oh, menopause causes this, or hormone therapy causes that. But I think we have to really be understanding that there are many kinds of menopauses. There are many kinds of hormone therapies, just like there are many kinds of hormonal contraceptives. So The combined pill seems to be less responsible for any kind of depressive symptoms in humans, and I think that's important information to move across. But the data that does seem to be out there, and the one that's coming to mind right now is that antidepressants in combination with hormone therapy perimenopause seem to be more effective than outside of that, if you're not taking hormone therapy. In terms of gender-affirming hormone therapy, I think it's, again, even less work done there. We looked at all grants for the last 12 years. It's published from CHR, and there were zero grants on transgender and gender-affirming hormone therapy on the brain, zero. So we don't know, but the data, the little bit of data that's out there from the US and other areas would suggest that some depression rates go up, but it goes up with male to female. It goes down maybe with male to female, but it's really hard to make conclusive statements about that. There's a lot of comorbidity with transgender too, as well, with psychiatric disorders, so we have to be a little careful about what changed and what didn't change. Yeah, you're welcome. I think it's a really good question. I really hope somebody starts to study it. Yes, I know it starts with a Z. Yes. You told me that you'd be happy if I remember that.
Zoria.
Zoria, thank you.
Yeah. So my question is, we talk a lot about like prevalence and onset and sex differences for depression in that regard. In terms of kind of intensity and reoccurrence, is there any literature to suggest that males or females experience kind of frequency of, if it's like a reoccurring depression syndrome, or in terms of intensity of the actual symptomology? Are there any differences in that regard, or has it been pretty consistent, or just not steady?
Oh, that's a really excellent question. I don't know if people heard it, but it was about reoccurrence and chronicity and severity, and do we see any sex differences in any of that? I have a thought in my head that may or may not be based on actual literature, so I'm hesitant to say it. But my best guess is that the greater severity is in females, greater chronic, not greater, but like more prevalent female chronicity, how would you say that? Greater reoccurrence, more frequency, yeah. But I don't know how well it's studied. Like I said, when I was looking for those sex differences in biomarkers and sex differences in symptoms, they literally were like 4 studies out there. Because when you PubMed it, and I've had reviewers do this and say, oh, well, I found 2,000 studies. No, that's when people said, we looked at males and females, or we looked at men and women, but they don't actually give you the data by sex. So if I don't have a table and a supplement or something, you can't answer the question. So unfortunately, there's a little bit, but not as much as we'd like. That's why we really do need to, even if you're not super interested in it, give it to me in a supplement. And hopefully there are other people that are interested, and we will look at it. So I hope I don't sound too preachy. Not every. You don't count, but you do count. You absolutely count. I don't mean it that way. I don't think that everyone needs to be like super passionate about it, but I do think we're gonna get a lot more answers if we start to pay more attention to it. So, like I said, supplement it for people that are interested.
But we do need somebody to be super passionate, and that's you, Liisa.
Yes.
Thank you so much.
Yep.
2 fantastic talks and so many thought-provoking discussions over the last 2 days. I know that you've changed what some people are going to be doing here. Yes.
Okay, thanks.
Please join us on the second floor lounge for more discussions with